Germination. Germination is the growth of seeds into seedlings. It starts when seeds become active below ground and ends when the first foliage leaves appear above ground. A seed consists of an embryo and its food storm surrounded by a testa seed coat. The embryo is made up of one or two cotyledons seed leaves attached to a central axis. The upper part of the axis consists of an epicotyle which has a plumule embryonic shoot at its tip. The lower part of the axis consists of a hypocotyle and a radical embryonic root. After dispersal from the parent plant, the seeds dehydrate and enter a period of dormancy. Following this dormant period, germination begins provided that the seeds have enough water, oxygen, warmth, and in some cases light. In the first stages of germination, the seed takes in water, the embryo starts to use its food stock and the radical swells, breaks through the testa and grows downwards. Germination then proceeds in one of two ways depending on the type of seed. In epigeal germination, the hypocotyle lengthens, pulling the plumule and its protective cotyledons out of the soil. In hypogeal germination, the cotyledons remain below ground and the epicotyle lengthens, pushing the plumule upwards. Hypogeal germination, broad bean, Vicia faba. Seed at start of germination. Testa seed coat, cotyledon seed leaf, plumule embryonic shoot, epicotyle upper part of axis, hypocotyle region between epicotyle and radical embryonic root. Shoot appears above soil. Lateral root, primary root, epicotyle upper part of axis lengthens. Young shoot, cataphyll, scale leaf of plumule, cotyledons, seed leaves remain within testa seed coat below soil's surface. Radicle breaks through testa. Split in testa seed coat due to expanding cotyledons. Testa seed coat, plumule embryonic shoot, hilum point of attachment to ovary, cortex, vascular tissue, xylem and phloem, epidermis, root tip, region of cell division, radical embryonic root. Hypocotyle, region between epicotyle and radical, epicotyle upper part of axis, cotyledon seed leaf. Foliage leaves appear. Lateral root system, primary root, cotyledons, seed leaves remain food source for the seedling. Cataphyll, scale leaf of plumule, epicotyle increases in length and turns green. Stipule structure at base of leaf, foliage leaf. Epigeal germination black bean, Phaseolus species. External view of seed at start of germination. Testa seed coat, raphe ridge, hilum point of attachment to ovary, micropyle pore for water absorption. Longitudinal section through seed at start of germination. Testa seed coat, cotyledon seed leaf, hilum point of attachment to ovary, radical embryonic root, hypocotyle region between epicotyle and radical, epicotyle upper part of axis, plumule embryonic shoot. Radical breaks through testa and lengthens. Root cap protective covering for root tip. Lateral root, hypocotyle region between epicotyle and radical. Cotyledon seed leaf, plumule embryonic shoot, testa seed coat, epicotyle upper part of axis. Hypocotyle hook appears above soil. Primary root, lateral root, hypocotyle region between epicotyle and radical elongates. Hypocotyle hook pushes out of soil. Testa seed coat splits. Hypocotyle straightens. pulling leaves and cotyledons out of soil. Testa, seed called shed. Hypocotyle region between epicotyle and radicle straightens and lengthens. Root, cotyledon, seed leaf. First foliage leaf protected by cotyledons, seed leaves. First foliage leaves fully developed. Root, testa, seed pod begins to disintegrate. Cotyledon seed leaf withers. Growing point, petiole leaf stalk. First foliage leaf fully grown.